Hi, everybody. It's noon here in Ligonier, and so we're going to go ahead and get started with the Natural Areas Association webinar on beech bark disease given by Dr. Connie Hausman of Cleveland Metro Parks. Thanks for joining us. We have over 120 people with us, which is great. My name's Sarah. I am the new program assistant with the Natural Areas Association. I'm just going to give you a little bit of information on a few slides about the Natural Areas Association and also a few of the upcoming events that we have, and then I'll hand the webinar over to Connie. Before I do that, though, there will be a question and answer session at the end of the presentation, if there's time permitting. Please go ahead and use the chat function in Zoom to test out typing questions in for that section. For now, go ahead and type in what motivated you to join the webinar today, whether it's an interest in plant pathology, or you're managing forests, or you're within the range of BLD as far as it's known. Great, some people are already typing in working in forest monitoring, forest ecology. In the chat window, make sure that the to field is to all panelists and attendees. Got quite a variety of people coming in. This is great. Continue to send those chats in. So just real briefly, some information about the Natural Areas Association. We're the organization that connects, supports, educates, and advocates for the national community of natural areas professionals. We're a 501c3 nonprofit, and we've been around for over 40 years. We provide up-to-date scientific information for natural areas management through webinars like this one, our annual Natural Areas Conference, regional workshops, and the Natural Areas Journal, which is a peer-reviewed quarterly publication. I encourage you to visit our website, naturalareas.org, to check out all the great things that we have going on, and connect with a colleague who's already a member of NAA. They're really our best ambassadors. We have some events coming up in the next few months. We've got 2 webinars. One is next Wednesday on spotted lanternfly, given by Sarah Wurzbacher. She's a Penn State forestry extension educator. That one will also be at noon Eastern time. We're also planning a webinar for November 13th with Vanessa Beecham from Townsend University. And then we've got our annual conference coming up October 8th through the 10th in Pittsburgh, Pennsylvania. So be sure to check all of those events out at our website, naturalareas.org. And now for the main event. Dr. Connie Hausman is the plant and restoration ecologist with Cleveland Metro Parks, and she's the executive director of the Ohio Biological Survey. She's with us to share up-to-date information on beech leaf disease. She's also an NAA member, and she'll be at the Natural Areas Conference in October, so we look forward to seeing her then. Once again, there will be a question and answer period at the end, time permitting. Go ahead and type your questions into the chat while Dr. Hausman is talking, and then I'll ask her those questions at the end as we have time. One other note, this webinar has been approved by the Society of American Foresters for one Category 1 continuing education credit. I'll be following up with an email after the webinar, so if you're interested in receiving continuing forestry education credit from the Society of American Foresters, please reply to that email. I'll be asking for your license details and so on. All right, time for Dr. Hausman to take over, and she's got great information for us.
Great, thank you so much, Sarah. I'm just switching screens now. Are we good? All right. Well, thank you everybody for taking your lunch hour to listen in to this webinar. There's a lot of information that I hope to be able to share today, and I'm going to try to go through it as much as I can quickly so that we have enough time at the end for various questions, as I will predominantly share information and data that we have in our park system, efforts and work that we've done nationally and in collaboration with several different partners. So this range and distribution map, if it's not familiar to you already, illustrates where American beech is currently found. Knowing this audience, I don't necessarily have to share too much information about what beech trees look like, but this is our area of concern as we're tracking and monitoring beech leaf disease. The first observation of BLD occurred in 2012 by a park biologist, John Pagachnik at Lake County Metro Parks. And it's found there just on the shores of Lake Erie. John is a great naturalist. He's a great biologist, and he's in the field constantly making observations and providing feedback to various regional partners. The shaded area that you see in purple is the glaciated Allegheny Plateau. That region I highlight explicitly because we happen to have a partnership of various conservation agencies across the LEAP region. And this is a consortium of over 45 different conservation-based member organizations. We meet bi-monthly as a way to network and information share. And I put this slide up to acknowledge their efforts and to really illustrate the fact that early on in the story of beech leaf disease, that organization and the mechanisms of our bi-monthly meetings were a way that we were early on discussing the spread of these signs and symptoms. So this is what we're talking about. These are our classic interveinal dark banding patterns that we see on beech leaves. I have a range here on the screen starting on the left. Those are fairly mild, showing symptoms. So you can see a single band to a few bands within some of the interveinal areas between those lateral veins. The middle shows a little bit more advanced with additional striping and banding pattern. And the leaf pictured on the right-hand side are advanced symptoms. So these are much darker in color. They are solid bands. They are thickened and leathery in texture. So I'm going to go through each of those a little bit more. As we've been tracking the progression of beech leaf disease, we break them into three different main condition categories. Condition one are our normal size leaves. They have no symptoms. You can see that in the lower picture on the left-hand side of that twig. They're pale green, they're thin, they're light. Leaf condition two, like I said, illustrates a little bit more of those interveinal dark bands that we see. They tend to be normal in size, but you can see a variety of one to a few banding pattern on that leaf surface area. Condition three tend to be the more advanced leaf stages. These leaves tend to be smaller in size. They have curled edges. They are either more than 2/3 of their leaf surface area is covered with this dark banding pattern, or they're completely solid, in which case they are really dark green. They are thickened, they are leathery in texture. They almost feel evergreen in texture. That's how thick they end up feeling. From a phenology perspective, one of the things we've noticed in the field symptoms, whether or not they are normal or asymptomatic, minor banding patterns or heavy, occur right at bud break. So the pictures that I currently just show on the left show right at early spring bud break. You can still see the bud scales there. And the zoomed in individual leaf at the bottom, you can start to see some of the crinkling and some of the dark interveinal bands. The image on the right, I laminated several different early developing leaves. Those illustrate in the bottom left portion of that picture a normal leaf that is asymptomatic, and then a variety of other leaves in different conditions. And so the two leaves at the top of that laminated sheet show the contrast between fairly mild symptoms with just a few bands, and then right next to it on the right-hand side, that thick and dark leathery leaf that is, again, first apparent right at bud break. So this is what all three of those conditions look like on a single branch. And so there's a few patterns that we've noticed with beech leaf disease. One, all of the leaves that develop within a single bud are very similar in symptom expression. And so the very center cluster of leaves, leaf condition 1, those are all normal and asymptomatic. If you look just to the right, you can see leaf condition 3. All of those leaves that emerge from that bud are very heavy in symptom, and those are dark green, smaller in size, thickened and leathery. So on a single branch, you can have clusters of asymptomatic leaves, minor symptom leaves with a few bands, or these heavily symptomatic leaves. And they are also variable in expression along that branch. So again, you can see from the image, you can have asymptomatic leaf clusters next to heavy symptoms, next to mild symptoms. One of the other things that occur over time is you end up having branches with aborted buds. And so these, the bud development on these twigs tend to be smaller in size. They're very dry and papery. They have no live tissue inside, although they persist on the stem typically through the growing season. And so you can find these aborted buds on twigs as they initiated some development, but then failed to flush out. And that ends up resulting in twig dieback and eventually branch dieback as well. So these are a couple of images looking up through the canopy in which you can see that dark interveinal banding pattern. The difficulty when you're looking for beech leaf disease early on is because those beech leaves are so thin that they cast shadows when you're looking up through the canopy. And so early symptoms in which you're looking at just a few bands can be mistaken for shadows and overlapping leaves. But here in these images, you can clearly see that those symptoms are quite evident as soon as you turn that search image on. From a field observation perspective, we're first detecting the presence of beech leaf disease in our seedling and sapling layer. So it occurs in the understory first, small shrub layer trees, sapling trees. By the time it gets to a large tree, it starts in the bottom portion of the canopy first. So again, the first signs and symptoms occur in the seedling and sapling layer. Once it matures into one of the mother trees or nearby, it starts in the lower portion of the canopy before it moves up to the upper portion of the canopy. I wanted to talk a little bit about funding that we received last year from the U.S. Forest Service through their emerging pest and pathogen funding in order to help facilitate some national survey efforts. It was clear that these signs and symptoms were spreading across the landscape, and because of our position in proximity to Lake County and the resources that we have, it made sense for us to try to help contribute to those national survey efforts and to work with our partners to establish and assess some monitoring plots, and again, track a little bit more rigorously the symptom expression of BLD throughout the growing season. So this is the current range and distribution map of our known BLD counties. Again, I want to reiterate that this map and this project is really a mechanism for us to track leaf symptoms. So these are observational leaf symptoms of that dark interveinal banding pattern. And so you can see Lake County is in that pale yellow color in 2012, and each of the county distribution maps are colored accordingly. And so in 2018, we ended up with 24 total counties in Ohio, Pennsylvania, 1 in New York, and as well as our partners in Ontario. That data project actually ended up having 12 different mechanisms for capturing and recording the presence of these leaf symptoms. And it was a bit cumbersome to try to manage all of that information. And so what we've modified and done for 2019 is really try to advocate for the use of an app So this is a bit of a spin-off app from a separate project that we had done in partnership with Kent State University. And this app is called Tree Health Survey. This is a mechanism for us to systematically survey various points on the landscape with state and local partners, citizen scientists, resource professionals, so that we can have a better understanding of where beech leaf disease occurs, but also ask a series of standardized questions so that we can make sure we're accurately capturing the severity of each of those locations as they're reported. It is currently only available on Apple devices. Our Android version is still pending. However, for those partners that don't have Apple devices, we have paper data records that we can share and disseminate, as well as a formatted Excel file that we're happy to share, so that all of the records that are being captured in the field ask the same standardized questions. So these are a couple of slides that just illustrate what that app is. It allows us to go through a small training module to make sure that people are identifying beech accurately, and that there is a series of pictures and trainings to identify who you are, where you are on the landscape, what the representative tree that you're collecting information from, and what those signs and symptoms look like. And again, within each of the app, you can find pictures to illustrate or to provide clarification on a symptom that you might be looking for. And in doing so, then you create a profile for yourself, so that as soon as a record is submitted, if we have questions or concerns or we'd like to follow up, we can contact you through the profile that you have created within the app. All of the survey information and data is being compiled by Dan Volk. He is our Forest Health Project Coordinator, and he is supported with that funding from the Forest Service to facilitate all of the records that are being collected on the ground across our various state partners and other agencies. So his name and contact information is there. We recognize that there are still limitations in this survey, as it is only available on Apple devices. And that mechanism also does require data to connect and submit a record. So our short-term resolution for that issue is to, again, provide those paper data sheets or the formatted Excel to be able to share, so that you still have a mechanism for collecting and capturing records here still in 2018. We just received notification that we did receive additional funding that will allow the developers at Kent State University to expand the functionality of this app, to create an Android version, and also allow data collection to occur while you're offline in the field. And that makes it a lot easier for those of you that work pretty remotely. And so that's still a little bit pending, but something we're looking into trying to develop and modify as soon as possible. So I wanted just to share this map because it gives you a sneak preview a little bit about what's going on for 2019's data in the state of Ohio only. So our goal or objective for Ohio was to try to collect about 10 records per county. Thus far, we have over 550 records submitted through the app, which is really fantastic. But you can see that there's still plenty of data gaps that we'd like to be able to capture outside of the known range of BLD. However, in 2019, with the information we have thus far, we are adding 2 additional counties. So we do now have positive records in both Lorain County and Erie County, Ohio. So there is some Western movement of BLD. And you'll also note this year as well, the black dots in the upper left-hand corner are in Lake and Ashtabula County. And we actually also have dead records in Cuyahoga County as well. So this is the first time that we're actually capturing trees that have died on the landscape through the app as well. So coming back to the large range and distribution map that we have, so I just wanted to illustrate some of the information we have initially for Ohio. And I did that because the ODNR Division of Forestry did put the app on all of their Apple devices and are contributing to those records. And so that's been fantastic for why we have 550 records for the state of Ohio. Each of the other known states, Pennsylvania, New York, and Ontario in Canada, have other mechanisms for their survey. They are still asking the same questions that we're asking in the field, but we won't have their records until probably more like the end of the season. However, for 2019, each of the known affected states are going to be adding counties. So I guess the short story is there will be additional counties added for the 2019 year in Ohio, Pennsylvania, New York, and Ontario. Additionally, we have a record positive that came to us from Connecticut. And so Southwest Connecticut and across the harbor on Long Island, we have positive BLD leaf symptoms detected. And so that represents quite a different location for our survey efforts. And there conceivably are some other suspect areas in other surrounding states. So no matter where you're located, it's never too early to start looking. So I wanted to shift gears a little bit and talk a bit about what we have done in Cleveland Metro Parks and some of the existing data and work that we have that we're trying to mine for specific beach-related information and spread. And so our park system was actually established in 1917. We're only a year younger than the national park system. And we protect and preserve about 24,000 acres across Cuyahoga County, parts of Medina County, a little bit into Lake Geauga and Lorain County as well. We're broken up into 18 different reservations that affectionately are known as the Emerald Necklace around downtown Cleveland. So you can see from that cartoon our distribution of our protected land. In 2010, we initiated a long-term vegetation monitoring project that we set up across our park system to help us understand what type of natural communities we have, where they're located, and what the condition of those natural areas are. And so that sampling design ends up being about 1 data point for every 50 acres of protected land. So that's the aerial that you see in the color dots. You don't have to worry about the color dots, the specific colors themselves. That just shows the sampling density. And so the design is for us to collect 100 data points a year for each of 4 years. And like I said, it was initiated in 2010. So we have actually 405 total vegetation plots. So they're spatially balanced across the park system in their entirety and within each year. And so looking at those records, we actually have 203 total plots that have beech trees present. And so in the little table that I have here, it shows their original sampling date between 10 and 13, and then their repeat sample between 2015 and 2018. And we have about between obviously 37 and 60 plots that are sampled within that time period. So we actually can go back and track our original beech population condition prior to beech leaf disease and track its spread in its 5-year repeat cycle. So that's the data I'd like to share with you next. All of those plots combined end up resulting in about 3,000 trees and just over 2,600 shrubs. So it's a lot of stems that we have information for. Because it's natural areas, I know many of you will be familiar with the Carolina Vegetation Survey method. I just share this to be able to illustrate the fact that this is our field protocol and the standard plot design that we have for most of our forested systems. So in the 5-year repeat, like I said, 405 total plots, 203 with beech. After that 5-year repeat survey in 2018, we had 78 plots, that had beech leaf disease. So about 38% of our total plots had the presence of BLD. What that looks like over time is that it originally was found in Lake County, which is there into the northeast corner occurred in our eastern reservations in 14. It spread through 2015, 2016, and 2017. So within those 4 years, it spread throughout the entire county. If we look at the by year, I think it's really important to recognize just how quickly, once BLD does arrive within a given area, how many plots are affected between these years. And so 2015 on the left, 20% of our plots were affected. 16 had 33%, 17 was 48. By 2018, 62% of our plots had a stem affected with BLD. And if we look at that in another way, that just sort of shows you that in 2013, we did not have beech leaf disease in our park system at all. It didn't occur until 2015. Affected locations and affected stands through 2018. So in the upper right-hand corner, you can that are illustrated there show you where the initial BLD sites were that John Pogachnik found. And then the red highlighted reservations were those in 2014. And so we can definitively say that we have seen a western and southern spread of beech leaf disease across our park system at about 13.6 to 17 kilometers per year. The open circles are those plots that have beech but no beech leaf disease as of yet. And the dark colored, the black dots are those with beech leaf disease. However, there's some information and curiosity about whether or not there's a much faster movement to the east than there is to the west or south. And part of that may be attributed to landscape features. The size and scale of protected land in Pennsylvania is much bigger. The proportion of beech maple forests and beech as a system is much higher. We have a much more urbanized and fragmented landscape within Ohio, an agricultural landscape as we move even further to the west and south in Ohio. So what does that look like from a stand decline then at each of these plots that do have beech leaf disease? This is an image from an original plot sampled in 2011. And this is that same exact plot sampled 5 years later. And so rather quickly you end up having that significant spread through that shrub layer. So I had mentioned that we have about 3,000 trees and 2,600 stems that are sampled in total. This is the initial sample across year on the x-axis and number of stems on the y. In their repeat sample, these are the affected stems within a given year. Of our trees, by the time 2018 rolled around, we had 86% of the stems affected by beech leaf disease. The shrub layers, similar. So again, it was like 30%, 37% in 2015 for each of those, and 83, 86% by 2018. So within a given stand, the number of stems that are affected at those locations is significantly spreading. So this is, again, I just want to highlight again, in 2018, since it's our most recent amount of data, 62% of the plots are affected with beech leaf disease. The stems at those plots, 92% of them are affected to some degree with beech leaf disease. So once beech leaf disease occurs in an area, the rate of spread within the stand is rather fast. The question about mortality, this is still a little precarious because we're fortunate enough to have very early data in the spread of beech leaf disease across our landscape. And in 2018, last year, when we recorded mortality, we had about a 7% mortality rate. So that's 96 stems out of the just over 1,300 total stems that were surveyed, beech stems that were surveyed in 2018. However, they were fairly small. They're less than 10 centimeters DBH, But the totality is that within 4 years, you have a 7% mortality rate of these seedling and sapling layer trees. In the heavy affected areas, 9 plots had mortality in total. 3 of those plots contained 86 dead stems. So the within stand spread and the within stand mortality rate is rather significant. And unfortunately, each of these affected plots and these affected stands, we have not come across trees that are not symptomatic. So we have no, for lack of a better term, resistant trees. There are no asymptomatic trees within affected stands. They all are affected to some degree. We have only seen an increased rate of spread, an increased decline in canopy cover. And so we have an expectation that mortality rate is only going to increase in rate and affect greater tree size. These 2 panoramic images just illustrate, some of these heavily affected beech stands and the small sapling and seedling layer trees that are affected the most and causing those mortality events. A subset of those vegetation monitoring plots were selected in 2015 for us to annually check. So there were 13 plots that I selected, and those were based on the number of stems that were affected within that plot. I wanted a range of BLD symptoms, those that were fairly low in severity to those that were fairly high. I wanted to target plots that had approximately 20 trees per plot, so that way we could fully see each individual tree and its entire canopy for proper assessment. So our sample size is 302 trees across those 13 plots. Their mean DBH is about 7.6 centimeters, and the range is from 0.3 to 72. So we do have some large trees in there, but by and large, they're fairly small trees. So this is a pretty hefty graph, and so I'm going to spend a little bit of time to be able to get through it. And so the y-axis is our proportion of normal leaf cover. So we're looking at the adjusted normal leaf proportion of the canopy, and that it's adjusted because we take into account what proportion of the canopy had dieback. The x-axis is actually years with BLD. Because of the earlier information that I presented about when beech leaf disease first occurred in each of our reservation, the timestamp of when BLD first arrived in a reservation it is strongly related to the rate of decline. And so 0 is the year prior to beech leaf disease occurring. Year 1 is when beech leaf disease arrived. 2, 3, and 4 is how long that symptom has been within that reservation. The cluster of graphs are different tree sizes. So the seedlings are from 0 to 2 inches, saplings are 2 to 4, trees are 4 to 8 inches, and large trees are greater than 8 inches in size. And again, these graphs summarize 302 total trees. And we tried to keep well, there's a higher number of seedling stems and about 50 to 60 trees in each of the sapling tree and large tree size categories. And so what you can see if you look at the seedling cluster of graphs is that the normal leaf cover drops within year 2 to about 50%. So as soon as beech leaf disease occurred in that reservation, in small seedlings, the second year after it arrived, about 50% of the normal leaves were still asymptomatic. And then it continues to decline in year 3 and year 4. As you increase in tree size, saplings 2 to 4 inches, you see that same 50% reduction in years 2 and 3 Those years 2 and 3 stayed fairly similar, but then again, by year 4, you had a huge drop in the proportion of normal leaves. The larger size trees seem to maintain higher proportion of normal leaves a little bit longer. But in the 4 to 8 inch size category, again, by year 4, you had a significant reduction in that normal leaf cover. The large trees are even slower yet. So there's a delay in the decline in the normal leaf cover that occurs on larger size trees. So again, this data is backing up the anecdotal information that we observed early on, in that beech leaf disease first occurs in that shrub layer, seedling layer first, and causes the most and the fastest decline. And then by the time it reaches larger size trees, it occurs in the lower portion of the canopy and moves upwards. So inevitably, a question that we always end up getting is, what do we do about beech leaf disease? What are our treatment options? And so because of the annual plot data that we initiated in 2015 and 2016, we were approached by Beech Trees with an interest in doing an experimental treatment on a phosphite product that they had used to treat other diseases experimentally, and they wanted to investigate its application in treating beech leaf disease or increasing tree vigor to slow the decline. And so the next slide I'm going to show you is a little bit of data that we have from that project. We picked one of our annual plots to be able to implement this project. And so this is just some preliminary data from a subset of those trees. It's about 1/3 of the total project. So again, the y-axis is the percent of canopy cover, if that's total canopy cover, total leaf cover. And then the x-axis is year. 2016, again, this was one of our annual survey plots. So that's data that I had for assessing total canopy cover in 2016. We were approached again by Davey and implemented those phosphite soil applications in the spring and summer of 2017. Those treatments were repeated again in 2018 and again this year as well. The condition assessments that we did on a per tree basis are done in September each year. And so in 2017, those 2 treatments with the phosphite product occurred right at leaf out and during the growing season. And then the tree condition assessment was done the 2nd week of September. And so you can start to see on this smaller subset of the project that there seems to be some difference and some differentiation between the control trees in that reddish color and the treatment trees in blue. So this project we are continuing, and our annual data collection projects are scheduled to begin the 2nd week of September. The next set of slides that I wanted to share actually take a step back from the work that we're immediately doing and start to illustrate and highlight some of the other research labs that are interested in beech leaf disease and trying to help us figure out what is it, where is it, what's causing it, what can we do about it. And so I reached out to each of our partners to provide a slide that we could share with the audience. So the first project I'm sharing with you is one that is being led by the Ohio State University. And they are looking into molecular microbial profiling of beech leaf disease. And they have separate funding from the USDA to explore that work. It is being led by Enrico Bonello and his graduate student, Carrie Ewing. And they have several different field sites within Lake Metro Parks, within Cleveland Metro Parks. They have sites in Pennsylvania and Columbus as well. And with the hope and goal to discover the causal agents of BLD by characterizing and comparing the communities of fungi, bacteria, phytoplasms, on symptomatic and asymptomatic leaves. So the next several slides, I'm going to be sharing with you these partner collaborated projects. And I've tried to leave names and email contacts up briefly so that everybody can reach out to each of these labs individually for follow-up questions or inquiry on their results. This next project is actually looking at the role and relationship of beech leaf disease with a recently discovered foliar nematode. And this work is in collaboration with Lynn Carta, who is with ARS, and David Burke at Holden Arboretum. There's a new citation at the very bottom, hopefully everybody can read. It's a recently submitted paper to Forest Pathology. And it is looking at beech leaf disease symptoms caused by a new subspecies, Littlelichnus crenati mccannii. In this project at Holden Arboretum, they took asymptomatic trees grown in a greenhouse, and they inoculated the buds with the Littlelichnus nematode that had been isolated and collected from within their collection. And during bud break, they were able to find that the new emerging leaves did have that dark interveinal banding pattern. So again, an asymptomatic tree inoculated with the nematodes, leafed out with leaves that did have that interveinal banding pattern. So that's a new research project that has recently been submitted to Forest Pathology. This foliar nematode question ends up being one of proportions and numbers and spread question. And so Lynn and another partner are looking at how that foliar nematode could be spreading within trees, between trees, and affect the range. And so they are investigating various invertebrates and focusing primarily on mites as potential vectors for the nematodes for short range spread. And so that's a little bit about their project as well. For our partners to the north, the Ontario Ministry of Natural Resources and Forestry is also determining the role of Littlelichnus crenati mccannii in BLD. They are developing new survey methods for detecting those adult nematodes with the hope of being able to help understand and implement other forest management options, as well as the relationship of beech leaf disease and beech bark disease, as both of those are currently present within their affected stands in Ontario. And Dr. Sharon Reed is leading that effort, and her contact information is there as well. So I know that was a lot of rapid fire information, and so I'd really like to acknowledge all of the partners across not only the LEAP region, but across all of the states. Our various state agencies and federal partners have been really great. I've been really impressed with the collaborative dialogue that's occurring in between Canada and the United States, as well as between the states. And I am especially grateful for the funding from the Forest Service to do this work as well. So with that, I'll take any questions, Sarah, if we're good.
We are good. We have 15 minutes for questions, and folks have been submitting them via chat and the Q&A. Connie, you actually covered quite a few of the questions about the disease biology, the vectoring, the causal organism. Just to pick up on that, there was a question about whether beech leaf disease is affecting any other beech species besides American beech, whether it affects tricolor beech, and whether it affects other genera of trees and shrubs.
So thus far, they have found beech leaf disease symptoms affecting several species of Fagus. So European beech, Fagus sylvatica. We had done survey work at Holden Arboretum through their collections, and they were able to find that dark interveinal banding pattern on European beech of various cultivars. Also the Korean beech, but not the Japanese beech. Fagus crenata does not, I believe, have the dark banding pattern.
Okay. And then we had a question about treatment, whether trunk injections of oxytetracycline would work. The participant said that it works for bacterial leaf scorch, so they're wondering if it would be useful for fighting beech leaf disease.
That's a good question. I know that both Davy Tree and Bartlett Tree, for that matter, have a couple of different projects in which they're experimentally looking at some treatment options. I can't speak specifically to that particular product.
Okay. And then we had a question about the tree health survey, whether there's any data sharing happening with EDD MAP-S.
So the EDMAPs, not specifically, although some of the different states use EDMAPs more than others. There definitely is a possibility for other data collection collaboration. We're still kind of in the early stages of trying to figure out the best way to make some of this information easily reportable and accessible. The 2018 data that we compiled for the range and distribution map that I shared, there were 12 different formats of data collection that we received and had to compile and consolidate and sort and standardize. And so the tree health survey was an opportunity because the platform and the citizen science project was already developed. Again, we had a partner relationship with Kent State University with funding in collaboration with Cuyahoga Valley National Park. There was an app called Northeast Ohio Park Apps, in which it was a collection of various citizen science projects. And the beech leaf disease one that was created within that, but because there seemed to be so much interest or access and ease for the reporting, tree health survey was spun out of that uniquely.
Okay. Two questions about the research on the pathology and how that might guide monitoring and surveys for beech leaf disease. The first is whether there are any particular stressors like drought that would predispose trees to beech leaf disease.
Good question. Whether or not there are other climatic stressors that have increased the vulnerability or allowed beech leaf disease to spread faster in one direction or with greater intensity or severity, that is conceivably possible. Early on in its detection, we could find no specific landscape feature or attribute about where beech leaf disease originally occurred. So it was on a forest edge, it was on a forest interior, it was a young forest, it was a mature virgin beech maple. And so then as other information came along and we were trying to look at various drought or rain events or harsh winters, nothing came up consistently as a pattern for us.
Okay. And the second question is whether there are any correlations or you suspect that there might be some correlations between human activity like recreation and beech leaf disease occurrence that would suggest that surveys should focus on campgrounds or day use areas or trails.
Right. Excellent question, especially in light of how the emerald ash borer spread around and the facilitation of infected firewood. That's a really hard thing to answer thus far. And I say that only because with the new information that we're receiving in 2019 survey reports with an occurrence now in Connecticut and Long Island, that's not, are those unique or separate introductions? Is that spread? Like I said, we have seen these on forest edges. We've seen them in forest interiors. It's in the Allegheny National Forest in fairly remote locations where people don't tend to go. And yet then there are other trail systems in which they are heavily campground oriented. Again, that could be an excellent question for someone else out there to help collaborate and ask.
That sounds like more research in the pipeline then. Another question about the tree physiology, the disease. Do infected trees recover, or do they flush new asymptomatic leaves after a twig has shown BLD symptoms? So they drop those leaves and then are they able to flush new foliage?
In the last couple of years, we have observed a late flush of leaves. It's not consistent, and it's not necessarily significant across the entire range, but we have noticed some of the trees have set out a late summer secondary flush of leaves. In that case, I have only ever observed them to be always asymptomatic. So unaffected, heavily affected BLD stems, that secondary flush of leaves have always been asymptomatic. They are small. Proportionally, they are very small in size, and they're very thin. They're very pale, they're very thin.
Okay. And then a question about collaborations. Are there any programs or research collaborations happening in Ontario with the Ministry of Natural Resources or Environment Canada that you know about or are involved in on beech leaf disease?
Dr. Sharon Reed is in Ontario, and we actually talk to and communicate and data share with them upon request. And so any time that we have the opportunity to collaborate, we will. Dan Volk, as a matter of fact, went to Canada to be able to help do some of the training about signs and symptoms that we see in our area versus the signs and symptoms that they have, and discuss what our monitoring program is so that we can standardize the additional field data collection efforts.
Okay, great. And then for folks who want to go out or are already out in the forest and will be able to kind of keep an eye open for beech leaf disease, who do they report symptoms to?
Well, the easiest and fastest way would be able to download the app because that will take a GPS location and allows you to take a picture of the tree that you're looking at and submit it immediately. And when you create that profile, then Daniel Volk, who's that forest health coordinator, can be immediately in touch to verify that site. He then contacts, if it's a new location, he then contacts the various state agencies to notify their either Department of Agriculture or the Department of Natural Resources to let them know that a new record in a new location has been found, so that then each state agency can send out someone else to confirm that location.
Okay, great. So for folks who are going to go out and do surveys, having access to that tree health survey app is the way to report occurrences where they are, no matter where they are, whether they're in the current known BLD range or beyond it.
Otherwise, they can email Dan at the address that I had provided earlier in the presentation and get a paper version, because again, that app only works if you have access to data. And so if you're in a remote area, then you would definitely need to collect it on a paper format. And so he'd be happy to share with you that mechanism.
And if they do collect the data with a paper format, you're probably going to want to see photographs and GPS coordinates and all that good stuff.
You got it.
All right. Just one other question about how the disease is vectored. There's some concern that we as humans moving through the landscape might be vectoring it, or that birds or other species would be carrying the causal agent of beech leaf disease, whether it's the nematode or another agent. Is that a cause for concern? Is there anything we can do to minimize that?
Right. So, you know, as we are exploring that role and relationship that the foliar nematode has in causing these signs and symptoms that we see on the landscape. That foliar nematode needs to be able to figure out a way to get from tree to tree and site to site. And so that's the additional work that they're looking on, those mites, those other invertebrates that might be carrying the nematodes, but then the mites themselves, maybe the mites themselves are then vectored by birds. So, you know, what role or relationship the wind, birds, mites, other migratory components through our forests, ourselves, are spreading this are still yet to be determined.
Here's another question. This is about whether trees with beech bark disease, trees that are resistant to beech bark disease have been exposed to BLD and what that looks like pathologically.
Yeah, excellent question. And that is part of the work that we're doing with the ministry in Ontario, as they have sites in the field that have both beech bark disease and beech leaf disease in the same location. And so we're trying to establish a set of monitoring plots that are currently asymptomatic for both, symptomatic for beech leaf disease, symptomatic for beech bark disease, and then symptomatic for both beech bark and beech leaf disease.
Okay. And then going back to the Davy phosphite treatments, 2 questions. How frequent were those treatments? And then whether you think there are other products other than soil treatments that might be useful to guard against beech leaf disease?
So their treatments were applied 2 times a year. They were applied in the spring and in the fall. And they were applied in 2017, 2018, and in 2019. And some of that work, we're going back out in 2 weeks to resurvey those trees. And in working with Anand, really trying to figure out how we can make that information and summarize that so that it's accessible and available to everybody is really important. I know that Bartlett Tree is also working with our partners at Holden Arboretum to do various trunk injections of, I believe they have a fungicide that they're doing a trunk injection. I believe they're also doing a nematicide so that they can treat those trees and track symptom expression and health over time. It's a little early yet. Other than that, unfortunately, there's really not management recommendations.
Okay, and then what was the thinking behind experimenting with the phosphite treatments to begin with? What was the reason for choosing that particular product?
So that's an excellent question that I would recommend following up directly with Davy about. So I don't want to misspeak too much. However, I can say that their interest in that product was because they had used it to treat some other tree diseases that had similar expression. I think there's a citrus greening that they had been using that product on, and they were interested in the impact of its use with some potential for antifungal properties, as well as essentially some fertilizer, since it's a phosphorus, to increase tree vigor.
Okay, great. Well, I think that we've pretty much covered the questions that came up on the content of the presentation. Thank you, Dr. Hausman, for a really informative presentation, and thank you for your time. Other people are chiming in. Excellent presentation, Dr. Hausman. Thank you very much. For everybody out there watching the webinar, I will send a follow-up email with a link to the video of the webinar, so you'll be able to look at Dr. Hausman's slides and actually hear the whole webinar again. I'll also include information on the Tree Health Survey app and the contact information for submitting paper records of BLD occurrence. Thanks, everybody. Have a good afternoon. And don't forget to register at the NAA website for next week's webinar and our conference coming up in October.
Thank you.